Hello, everyone. Welcome to our fifth Fellows Science session, where we will be hearing from our fellows and their research related to microbes and viruses. My name is Corrine Jackman Burden, a postdoctoral fellow from Carnegie Mellon University. I will be moderating this session along with my colleague, Dr. Sunny Narayanan, a research faculty at Florida State University. We are happy to be here today to highlight the amazing research of our fellows, and we appreciate your time and support in joining us today. Each fellow's presentation will be 15 minutes, followed by 5 minutes for Q&A. Fellows will be notified when 2 minutes are remaining for their talk. At any time during their presentations, please post your questions in the Q&A box on your screen. Since time is limited, it likely be that we won't be able to ask everyone's question, so please try to keep your questions succinct. The moderators may paraphrase and/or combine your questions if we observe a theme. So without further ado, I would like to direct our attention to the first fellow who will be presenting today. First, we will have Dr. Corey White. He is an NIH Blueprint D-SPAN postdoctoral research fellow in biochemistry at Johns Hopkins University School of Medicine. The title of his talk is Evaluating the Impact of SIV Infection and ARTS on the Spatial Distribution of Brain Lipids.
Yes, thank you so much for the kind introduction. And also thank you to all the organizers for the opportunity to speak with you here today. So let me just pull up the slide. So today I'm actually going to tell you about my journey where it's really looking to understand more about brain lipid metabolism and how that goes for a number of different questions in different contexts. So really from understanding the basics of how brain lipids are modulated and consumed to, for my postdoc, how within infection, how they're spatially changed by HIV, and moving towards independence, how social determinants of health impact brain lipid metabolism. So firstly, let me tell you why brain lipids are important. Your brain is actually your second most fatty tissue. Only adipose tissue is more fatty than the brain. And the manner in which the brain is comprised, those lipids tend to be longer within the membrane phospholipids within the brain. So those fatty acid tails tend to be longer, and there also tend to be a higher degree of unsaturation. Now, some of these lipids are also very necessary for proper neurodevelopment, including DHA, which tends to be something many people that have been pregnant are aware of. Additionally, the way that these are maintained is pretty well regulated. So a component of my thesis research was looking at a pretty novel enzyme and seeing how that is modulated, and loss of this enzyme that controls phospholipid head group actually shifted the balance of phospholipids. And this enzyme is quite unique to the brain. And in addition to just that regulation and that modulation, brain lipids are also consumed at a higher proportion than often previously considered. And normally when you consider consumption, people tend to think of glucose and ketone bodies under starvation. However, another major component of my dissertation was really being the first to uncover that endogenous fatty acids are being mobilized for their oxidation in the mammalian nervous system. And this was first just validated functionally through radio-labeled fatty acids. And there was a loss of that consumption. So there was carbon dioxide here that was generated that's radio-labeled for the wild type, and the knockout in gray is really just presenting that there's a loss of that carbon dioxide being generated, and that's phenocopied with etomoxia. So additionally, from the in vivo model, with the loss of this obligate enzyme for fatty acid oxidation, what was really compelling was there's a major increase in a fatty acid oxidation intermediate known as the acylcarnitine under normal conditions. So regardless of whether it's fed or fast, this accumulation occurred, showing that these fatty acids are being mobilized to be broken down, but they just can't due to the knockout model. And that just shows it's happening under normal conditions. So from my dissertation research, I've learned a lot about just how brain metabolism is occurring under normal contexts. And for brain metabolism at large, so expanded upon lipids, we know that other factors such as age injury and viral infections can alter the metabolism within the brain. So for my K00 phase, for my postdoctoral research, I designed a project with my postdoctoral advisor, Deanna Williams, to really look to see how brain lipid metabolisms alter by HIV infection. And brain lipids have been not well explored within the context of HIV. Now, HIV is very relevant to the brain. So the brain can actually be infected within the first couple of weeks of viral infection. So you have monocytes that cross the blood-brain barrier. They're able to activate astrocytes and infect astrocytes. And upon this infection and activation, the metabolic coupling that can occur between astrocytes and neurons has proposed to have been perturbed. Now, neurons, interestingly, cannot be infected, but they degrade to a high extent, which is hypothesized to be due to this loss of this metabolic coupling, as well as other factors, including increased chronic inflammation. Now, what is known is that in vitro, there's overexpression of the viral protein TAT, which increases fatty acid oxidation genes, as well as overexpression of, as well as elevated oxygen consumption within the brain, well, within those primary cells. Additionally, to just infection itself, the antiretroviral therapies used for HIV, used to treat HIV infection, have their own metabolic impact. So this occurs both in a cell type manner, as well as they have their own impact based on drug. And some data that are generated just validates this. So both for just basal respiration and for maximal respiration, there's elevation in oxygen consumption observed when treated with dolutegravir, a very common integrase inhibitor. However, the brain itself is very spatially heterogeneous, and this also is very applicable to HIV. So we know that brain atrophy is associated with HIV infection, and this occurs in very distinct regions. So basal ganglia, corpus callosum are two regions that are very much so predisposed to be damaged. And additionally, what is known about expression of a lot of brain lipid metabolism enzymes, as well as some ex vivo research, has shown that different regions of the brain at least have the capacity, just through those ex vivo studies, for differences in metabolic capacity. So hippocampus has been shown to have higher expression of fatty acid oxidation enzymes than cortex. So, and this need for more spatial imaging in the context of HIV has been presented within a small editorial comment that I had the opportunity to write for the AIDS Journal. So the question I really want to get to you and present to you today is how are brain lipids altered due to HIV infection and antiretroviral therapies? And to do so, we're spatially assessing this in a model of HIV infection using MALDI imaging mass spectrometry. So to do so, I have a piece of a tissue from a non-human primate model of HIV. Matrix is applied to help ionization. And upon ionizing, we have a full spectrum, which from these data, the laser comes across at every 50 microns. So we have one of these spectra for every single one of these points. And from these data, we can really look to see for individual peaks, what is the heat map for how this presents on a slide? So how abundant is this particular ion in different places for our slice? And cooler colors represent less, warmer colors represent more. And I've been able to do this for mass charge ions between 100 and 1,000 for a range of 591 ions of interest. But there's more that can be discerned. So bringing this capability to the lab, I merged this with a model of non-human primates that we have within our laboratory. And we have a couple of different groups. So we have non-human primates or that are rhesus macaques that are unaffected and untreated, those that are treated with SIV only, or simian immunodeficiency virus, is basically an ortholog of HIV for macaques or for non-human primates. There's also those that are treated with HIV, that are infected with SIV, and treated with the antiretroviral, so they're virally suppressed, a model of what canonically happens within people, as well as there are those that have been infected, but then had antiretrovirals withdrawn. And I have a preprint that assesses just the variation within this virally suppressed group, but I'm going to share a little bit of the capabilities of what can be done with some new data. So throughout this, I can put basically a piece of tissue from each group on a singular slide to really prevent any batch effects and allow for direct comparison, which makes this a very powerful tool. And just as an example of how these data go, we can see for these two acylcarnitines, the type of lipid that I mentioned earlier, just differences in the distribution that occurs between some of these two. And that can be represented and analyzed and have ongoing analyses to really look across so many of these ions. But one thing I really want to share with you today is that we can also do very broad analyses. So we have a principal component analysis that really looks to see what is the basic comparison between these two, between components. And we can see here that the unaffected is most similar. So colors that are more similar to each other are more similar. So unaffected is most similar to SIV plus ART and less to the SIV and ART withdrawn. So in conclusion, we know that MALDI is a very powerful method, and the initial analyses from these data suggest that ART treatment is bringing things more similar to the uninfected. And these ongoing analyses will allow for more powerful comparisons. But in the remaining time, I want to share what I would do for my own research program, which really looks to assess how social determinants of health are impacting brain metabolism in the context of neurologically relevant diseases. So, oops, I froze. HIV does not exist within a vacuum, or really no diseases exist within a vacuum. And there are multiple factors that can impact and contribute to health outcomes. And one thing that I'm doing now is assessing how stress, racism, and discrimination is impacting outcomes on HIV. So African Americans exist where they have higher indices of brain atrophy from HIV. So I'm looking for global metabolomics and really assessing the abundance of metabolic enzymes for pilot data for K99. And additionally, this can be expanded to other neurovirulent diseases such as COVID, or even other diseases that are not necessarily viruses, so that it also impact the brain, such as MS. And looking at how the intersection of any one of these factors is impacting brain health in the context of a disease. And this will be a multi-model system using bank tissue and within very well-controlled mouse studies, which will focus on two main aims, both of assisting of determining the brain lipidome and for assessing regulation and consumption of brain lipids, which will merge all of the techniques that I've gained along my journey to answer a number of questions and look at very specific things such as spatial changes of lipids, validation and discovery of new lipids, as well as evaluation of function, the temporal metric of oxidative metabolism, as well as an array of classic biochemistry techniques. And with that, I want to thank everyone that contributed to this work, especially the Williams Lab, and I'll take any questions. So thank you for your attention.
Dr. Ward, thank you so much for your presentation. While we wait for questions from our audience, I have a question for you. So you really, thank you for sharing your incredible research on so many different topics. One question I had was if there may be potentially adaptations with a vascular system in the brain, with regards to your observations of metabolic adaptation linked to neurological adaptations, with the vascular system providing oxygen, nutrients to serve metabolic processes in the brain. Would you speculate there are potentially vascular adaptations in line with your observations?
Oh yeah, so for this setup don't have the expertise in really addressing those very specific questions of how the vascular system has changed, but it definitely plays a role. So even with HIV, there are major changes to vascular integrity, and that contributes to the outcomes that are observed. So I expect that would also be the case with a number of other disorders, especially those that are impacted by levels of chronic inflammation. So tools to explore that, there are tools to map brain vasculature, and those would be quite useful to learn or to collaborate with others to add to this program.
Thank you for your response. While we're waiting for questions from our audience, I have another question. Since you mentioned the immune system, would you predict there are specific innate or adaptive immune cells involved with these adaptations that you're seeing, glial cells or perhaps T cells in the brain?
So the landscape of how different immune cells are interplaying might vary just disease to disease, but for HIV in particular, it is actually within the brain infected monocytes that are the key players that are coming in first. And HIV, interestingly, also does not infect neurons. So exactly why that's the case, at least I'm not well aware, and it seems to be like something that is ongoing for further characterization. But for the brain itself, at least in the context of HIV, it is monocytes and myeloid cells as a whole that are the major players.
Fascinating. Thank you so much for sharing. So we have a few questions from our audience. So one being, have you examined the impact of ART only treatment on metabolism in the brain with your SIV model?
So that is a wonderful question. We actually just had our first round of ART only macaques, and we've just collected samples from those. So that is in the works.
Fantastic. We look forward to seeing your future research on that topic. Another question being, if it's possible to alter brain lipids preemptively to protect from infection-mediated dysregulation, and relatedly, how much brain lipid variation is there exists between people and macaques, generally speaking?
So between people and macaques, that is also an interesting question to assess. I don't know of a study where there's been direct comparison with disease models. Since I do now have this human tissue, this is something I've really just acquired within the last month to do these. So these studies are ongoing, but that would be something I could easily incorporate as I'm doing analyses and just batch things together so things can be compared more directly.
Fantastic. A different question being if you have done any in vitro or in vivo work evaluating redox status following drug treatment, and related with that, if there may be any potential relevance for antioxidant treatment.
Ooh, I have not, but that is a good idea, just to see what are differences in redox status. I've not focused that much on that, but that'll be really interesting to see.
Fantastic. Thank you so much for your time and for sharing.
Oh, yeah, thank you.
Okay, thank you, Dr. White, for that very interesting talk. Now we will have our next speaker, who is Dr. David Anderson III. He is a postdoctoral research fellow through the NIH T32 grant in immunobiology at Washington University in St. Louis School of Medicine. The title of his talk today is MYC family members are dynamically regulated to control dendritic cell development and function.
Thank you for the kind introduction, and thank you everyone for participating in this symposium. I'm really looking forward to sharing my research and networking with everyone. Let me share my screen. Go ahead and get started. So I've made my title a little bit more concise in this version. So I'll discuss the epigenomics of immune cell development and function, and in the context of immune responses to infection as well as oncogenesis. And so I'm an immunologist, currently working in the laboratory of Dr. Kenneth Murphy, and also got my PhD at WashU in the immunology department. So my work has largely focused on the development and function of myeloid cells of the immune system, specifically dendritic cells, which are an essential subset of the innate immune system that develops from bone marrow progenitors to give rise to three major subsets known as classical dendritic cells, type 1 and 2, as well as plasmacytoid dendritic cells. These cells emerge from the bone marrow, circulate throughout the vasculature, and enter tissues where they survey antigens and detect pathogens. On detection of pathogens, they can mount specific immune responses as they migrate into lymphoid organs, where they process antigens from those pathogens or immunologically activating conditions and prime antigen-specific CD8 and CD4 T cells to mediate the adaptive immune response. And so working in the Murphy lab, which has really been an exciting time over the last several years, we've described how transcription factors regulate dendritic cell development, and in turn have used transcription factor knockout models in mice to delineate the specific functions of dendritic cell subsets. So in the context of type 1 dendritic cells, we know that BATF3, ID2, NFIL3, IRF8, for example, are transcription factors required for their development. And so when we knock those genes out in mice, we can attribute the functions of dendritic cells to deficiencies in specific intracellular pathogens like viruses and bacteria, the ability to mount appropriate immune responses during vaccination and with different adjuvants. It's really become clear now that dendritic cells are essential in mediating anti-tumor immune responses, and so have been really essential in mediating the efficacy of diverse immunotherapies. And so my work most recently has focused on a family of transcription factors, which we call the MYC family of transcription factors. There are three members, MYC, NMYC, or MYC-N, and MYC-L. They're known as proto-oncogenes, meaning they support oncogenic transformation. And this has been a very long-studied family of factors, maybe among the most important transcription factors discovered to date in terms of the length of research that has stemmed from those discoveries, dating back to as many as 40 years ago. And all of the MYC family members are known to be associated with different types of solid and liquid cancers in the case of leukemia. And so their roles have been studied at the molecular level in terms of their structure and function, but that work has mostly been focused on the member MYC and sort of assumed that those functions can be translated to the other members, although most these haven't really been studies that have been undertaken in all of the respective contexts. And so MYC and the family members have conserved DNA binding domains, BH, LH, leucine, zipper domains, and conserved transactivation domains, which are required to activate transcription and promote levels of transcription by RNA polymerase II across the genome. And so at low levels of MYC, You have MYC binding to promoters and activating gene expression, but at high levels, especially in super physiological levels like in cancers, large amounts of MYC can bind to promoters and distal enhancers to activate gene expression by promoting more rapid initiation of transcription by RNA polymerase 2. And so in the context of immune cell development in the bone marrow, we know that the different MYC family members actually switch between each other. And so in stem cells, like the hematopoietic stem cell, we have high expression of MYC-N, which undergoes a transition to MYC in myeloid and lymphoid progenitors that are rapidly proliferating to supply the entire blood system with immune cells. And then once differentiation occurs in the myeloid compartment, specifically with dendritic cells in mice and in monocytes in humans, this third transition between MYC and L-MYC occurs. And so we've been studying L-MYC in the context of dendritic cells and initially observed that L-MYC is expressed sort of across the board in all dendritic cell subsites, but it's most highly expressed by the type 1 dendritic cells in different lymphoid organs like the spleen, the mesenteric lymph node, mediastinal lymph node, and lung. And its expression is about a log-fold higher than other dendritic cell subsets. And so, our initial work in the periphery focused on this particular subset in mind. And so, because MYC family members activate transcription across the genome, it's thought that rather than activating a specific functional program, they're really supporting the basic cellular functions in whatever context we're studying. So cell growth, proliferation, DNA replication, RNA synthesis, protein synthesis, and all of that translates to a canonical phenotype among the MYC family members, which is that MYC deficiency, in this case L-MYC deficiency in dendritic cells, results in significant reduction in cell size. And so you can imagine reducing the cell size by twofold will lead to pretty significant reductions in their functional capacity. And so when we test the function of DC1s in L-MYC deficient mice, we find that in the context of listeria infection, for example, there's a significant reduction in the ability of dendritic cells to prime and activate functional CD8 T cells. At the molecular level, we wanted to ask, is it true that L-MYC is functioning like MYC? Certainly, the cellular phenotype of reduced cell size is conserved, but in terms of the genes that are being activated, does it have a specific immunological function, or is it, like I said, a sort of broad genome-wide support of biosynthesis? And so looking at dendritic cells in vivo, we saw, again, across the board that they're in L-MYC deficient mice, there's a broad reduction in the levels of transcripts associated with core biological functions like ribosome biogenesis, RNA synthesis and transcription, cell division. And this we found to be independent of the activation status of the cells. So in a healthy, unstimulated context, of an uninfected mouse, this is sort of the results that we see: broad reductions in transcription. And that we actually found that, this is really similar when we activate systemically with an adjuvant in vivo. And so what this is telling us is that although L-MYC expression is restricted to dendritic cells, which are important immune cells, it's not a master regulator of immune responses. It's really just a master regulator of housekeeping genes or biosynthesis. And so its immunological importance is attributed by the fact that it's expressed specifically by an important immune cell subset. And so studying gene regulatory networks has really sort of been what drives my interest in immunology and using the strong models in vivo that we have to test the function of genes during immune challenges or immunopathologies. And so one of the questions I asked was, well, what is the the regulatory mechanism by which these MYC family members are transitioning during dendritic cell development? And so we have broad and a pretty strong database of histone, lysine acetylase data sets where we can identify active enhancers across the genome. So if you just look at this particular panel, we have a CMOP, which is a monocyte progenitor. There's very little red indicating there's very little sequencing of pull down active enhancers. And so this is the LMYC locus, and we can see that the CMOP, the MDP, the CDP, these are DC progenitors, don't really have an enhancer in this region, but as soon as DC1s specify to the dendritic cell lineage in the bone marrow, we see strong activation of an enhancer here. And this corresponds to a direct binding of IRF8 indicated by ChIP-seq, and another factor, PU1, which can interact with IRF8 to activate transcription. So we have multiple lines of evidence suggesting that this IRF8 and PU1 factors are regulating the induction of enhancers during dendritic cell development, and other lines of evidence to support the fact that this transition is occurring in vivo in the bone marrow during dendritic cell specification. We have single cell RNA-seq showing that CMYC and LMYC in the common dendritic cell progenitor are mutually exclusive in their expression patterns. And so even in this what was thought to be a homogeneous population of dendritic cell progenitors, we're seeing the activation of LMYC and the repression of MYC, suggesting that there's a stage-specific induction of LMYC that corresponds to the development and differentiation of dendritic cells. And we can study, where these enhancers are and what the prevailing motifs or dominant motifs of DNA are in those locations and the putative factors that are controlling expression of the distal genes. And to confirm that IRF8 was driving LMYC expression in vivo, we looked at in vivo splenic PDCs or plasmacytoid DCs, and these again express LMYC highly in vivo in the spleen, but in the absence of IRF8 and IRF8 knockout mice, we see a severe reduction in LMYC expression, supporting our hypothesis that IRF8 drives LMYC expression. And to test whether the interaction with PU1 was also important in activating LMYC expression, we overexpressed an IRF8 mutant R294C on to the LMYC GFP reporter and IRF8 deficient background. And so we isolate progenitor cells and differentiate them in vitro into dendritic cells. So this represents sort of in vitro primary DCs infected with these retroviral overexpression constructs. And so we can rescue our LMYC expression in this genetic background by returning IRF8 to the cells. But if IRF8 has this single point mutation that confers, that limits its ability to interact with PU1, we ablate the ability to rescue LMYC expression, demonstrating that IRF8 and PU1 are acting together to support LMYC expression. And then to ask, well, what are the targets of IRF8 in PDCs? We found that IRF8 deficient PDCs fail to exit the cell cycle, and there's a large set of genes associated with control of cell cycle that are upregulated, suggesting that IRF8 is really essential in controlling this terminal differentiation stage where we see the transition to LMYC. And the next question was, well, what's controlling the expression of MYC in progenitors? And so we looked at the similar data set with histone, lysine acetylation, indicating really active enhancers in the MYC locus, 1.8 megabases away in the blood enhancer cluster. And at the same stage that we saw for LMYC, where DC1s are being specified in the bone marrow, we see complete loss of this enhancer, very potent and rapid loss of these active enhancer marks. And those also, interestingly, corresponded with binding of IRF8 and loss of accessibility in the chromatin, in this chromatin region, suggesting that IRF8 could also be controlling the repression of MYC. Again, by repressing the enhancer through direct binding and correlations with inaccessibility. And to test this in vivo, we crossed IRF8 deficient mice to MYC GFP reporter. And in common lymphoid progenitors and common dendritic cell progenitors, we saw a really severe defect in the ability of these cells to repress c-MYC or MYC expression. And so, we have this working model of IRF8 repressing MYC at the same time that it induces L-MYC. And IRF8 has long been of interest because it's associated with myeloid proliferation and is lost in some human AMLs. And so we have this system where human monocytes and DCs require IRF8 for differentiation. But we know that IRF8 represses MYC, limiting its the ability to use this factor to support oncogenesis. But in lineages where IRF8 can activate L-MYC, there's a possibility that MYC can be replaced by L-MYC in these AML clones or leukemic clones that also require IRF8 for development. And looking at cohorts of patients of AML, there indeed are IRF8 L-MYC high fractions of patients exhibiting high monocyte burden, suggesting that this gene regulatory network that we uncovered in mice is also acting in humans. And so the next stages for our for developing an independent research program is to continue to develop our model of IRF8 dependent MYC repression. IRF8 is a canonical activator, so we need to figure out how it is that IRF8 is capable of repressing a gene. We have a hypothesis that it is potentially interfering with the long-range regulatory sort of chromatin structure and interaction of promoters with enhancers. But it's also possible that there are novel interactions between IRF8 and co-repressors. And then we can translate these discoveries to human myeloid leukemias, where some of the cell lines that are already used in the study of AML show these patterns of IRF8 and MYC expression. We can test the ability or whether certain non-coding regions are required for expression using CRISPR-Cas9. We can develop myeloid progenitors in vitro from human cord blood or bone marrow, and then test the ability to interfere with oncogenesis by targeting regulatory regions that we identify through novel gene therapy modalities. And so this sort of is just a broader picture of using all of the methods that I've sort of worked on to date to define gene regulatory networks, and then use the immune system or our broad knowledge of immunological processes to validate whether these non-coding regions of the genome are important in immune pathology, vaccination, tumor rejection, etc. And so with that, I'll finish the talk. These are just some relevant works detailing some of my experience in becoming an immunologist and some of the awards and the PIs that have supported my work so far. With that, I'll take any questions. Thank you for your time.
Thank you, Dr. Anderson, for your insightful presentation. We have, we're going to be a little bit short on Q&A for you, but we have one, so start the question from our audience. What molecular interactions recruit MYC-L to gene promoters, and does it involve specific DNA binding sequences?
Yes, so the MYC family is part of a group of transcription factors known as EBOX binding factors. So these are DNA, they bind to DNA motifs together with a cofactor known as MAX as a heterodimer. And so those motifs are 6 base pairs of CAC, GTG. And so those, once they bind to those regions, they interact with transcriptional activators, that are potent at recruiting factors with enzymatic function, like histone demethylases and acetylases to regulate transcription at the promoter and also with RNA polymerase II.
Thank you for sharing. And so I have a question with regards to, so your presentation focused on dendritic lineage and biochemical genetic processes involved with those. At the beginning of your presentation, you highlighted some organoid or lymphoid organ distribution and the involvement with dendritic cells with those organs. So I'm curious from a cardiovascular perspective, specifically lymphatic physiology, how the distribution of these different dendritic cell populations may be tied with their transport by lymphatics to different lymphoid organs and different organs in general.
Yeah, so I think the prevailing sort of consensus from all of the literature combined and all of the contexts of lymphatic migration and circulation, we can maybe say dendritic cells are really specialized at exiting circulation. So if you take peripheral blood sample, they're very rare. And that's because as soon as they emerge from the bone marrow, they leave the circulation into all of our tissues. And then once they've sort of sampled the tissues for a while of antigen, maybe they've received a signal to become activated at steady state, which allows them to be sort of more of a tolerogenic immune cell that doesn't activate an immune response, or in the context of an infection, they would then sort of migrate through the lymphatics to lymph nodes where they would activate antigen-specific T cells. And that's sort of a process that would be occurring at steady state, a constant stream of dendritic cells from the bone marrow into the tissues, and then back into the lymphoid organs.
Thank you, Dr. Anderson, for your clarification and answering our questions.
Sunny, I think we have another question.
Okay.
Would you like me to ask it?
Question.
Certainly, I wasn't sure how we're on time.
I think we're good. We're okay.
Yes, one other question is if you have explored how MYC inhibitors affected the role and with dendritic cells.
Yeah, there is a small molecule MYC inhibitor. Traditionally, transcription factors have been difficult to target with small molecules because they usually don't have an enzymatic function, and their interactions with DNA aren't really that strong in terms of affinity. And so their interactions can be highly dynamic and transient. And so I guess in that respect, we haven't really studied MYC inhibitors in the context of dendritic cells. But also, an interesting observation that I'll just put out there is that dendritic cells are maybe one of the very few immune cell subsets that don't develop into cancer. So, we have T cell, B cell cancers, or, AML, all types of AML that, might be neutrophil dominant or monocyte dominant. You don't see that for dendritic cells. There haven't been any really strong reports, and maybe there's one plasmacytoid dendritic cell derived cancer, but those are extremely rare. And so there's something about proliferation and differentiation that's intrinsic to dendritic cells that's really incompatible with sort of immortality or and oncogenesis.
Thank you, Dr. Anderson, for your interesting presentation. Okay, so now I would like to introduce Dr. Guillaume Urteco. He is a Hannah H. Gray postdoctoral fellow in microbiology at Columbia University and Irving Medical Center. And today he will be presenting his research, which is spatiotemporal dynamics during niche remodeling by supercolonizing bacteria in the mammalian gut.
Hi, everyone. Thank you so much for the introduction. As you will soon see or immediately see, my title has changed. But that's because what I wanted to do today was to share with you my story of the insights that I gained from my postdoctoral work and how they've influenced my future research plans, which are to develop high throughput approaches to engineer microbial communities in the gut. So growing up and in graduate school, I spent a lot of time in Los Angeles. And for those of you who don't know, besides Hollywood and traffic, LA is known for having one of the most vibrant food truck scenes in all the US, where you can find just about any delicious or bizarre meal you can imagine. But the problem with all these eating fads that we see is that oftentimes we pay a lot more attention to this rating and we don't pay enough to this other rating. And unfortunately, this came to haunt a friend of mine. He ate at the wrong food truck, where his food was contaminated with pathogenic E. coli. And even after clearing out the infection with antibiotics, he's never been the same. And that's because the combination of infection and antibiotics had interfered with the balance of microbial communities inside his gut, a phenomenon often referred to as a dysbiotic microbiome. In his case, this perturbation led to a FODMAP disorder, where an excess of fermenting microbes in the gut had made him intolerant to many diverse sugars. He can't eat lactose products. He can't eat fruits because of the fructose inside them. He can't even eat garlic. Can you imagine that? Just terrible. And this is just one of the many disorders that can manifest when good microbiomes go bad. IBS, cardiovascular disease, diabetes, and even some neuropathic disorders have all been connected to this gut dysbiosis. And unfortunately, for how easy it is to perturb these communities, we're not very good at returning them to a healthy state. So in my postdoctoral work, I studied the primary therapy that is used to treat gut dysbiosis, fecal microbiota transplantation. In these therapies, stool from healthy individuals is transplanted into the guts of individuals with dysbiotic microbiomes. And in doing so, clinicians hope to transfer this healthy microbiome to engraft into these otherwise sick hosts. Very logical, right? So logical that it's been around since the 4th century AD, where Ji-E Hong, in his book Handy Therapies for Emergencies, described the formulation of yellow soup made from this healthy stool in order to treat diarrhea. But if that sounds gross to you, fortunately, we've made a lot of progress since then, and FMT is commonly used as an FDA-approved therapy for recurrent C. diff infection. However, there's anecdotal evidence that FMT could be used for so much more. In one case, a woman who received FMT for her C. diff infection also found that it treated her celiac disease. And there's even speculation that we may be able to receive FMT from top athletes in order to take our game to that next level. However, while there's lots of potential for these therapies, they have mixed success where only certain donors can transplant into certain individuals. And like in blood donations, there appears to be some logic to donor-recipient compatibility. However, we don't know the mechanisms underlying this logic. And even more interestingly, there have been documented cases of individuals with super donor microbiomes that can robustly transplant into a wide range of recipients. So for my postdoctoral work, I was interested in these two outstanding questions that will empower FMT, the success of FMTs. Firstly, why does FMT work in some cases but not others? And secondly, what are the characteristics of a super donor microbial community? And our approach to this was to use a mouse model to study what is happening to these microbes after FMT inside the recipient. And the reason this mouse model is so valuable is because we can control for diet, we can control for genetics, and we can also control for differences in immune factors that also play a role in colonization. And this allows us to isolate the role that microbiomes and microbes play in allowing for colonization by FMT. So despite controlling for all these other variables, you may be surprised to learn that laboratory mouse microbiomes are extraordinarily diverse. So to start this project, we acquired genetically identical mice from 4 different vendors, Taconic, Jackson, Charles River, and Envigo, and we sequenced their microbiomes. And as you can see, there's tremendous variability, not only at the phylum level, or at the family level, but at the phylum level, which this is problematic for some mouse studies because you don't know what microbiome they started with. But it's really exciting for us because it gave us the opportunity to study FMT outcomes between very diverse microbiomes. And fortunately, performing FMT in mice is extremely easy. All you have to do is put 2 mice in the same cage, and they will eat each other's fecal droppings. And in doing so, transfer their gut or colonic microbiomes between each other. So to do FMT between all of these different mouse microbiomes, we performed every possible co-housing. And then we sequenced the mice after co-housing to see what microbiomes were able to transplant in others. And here we're summarizing the results where we're showing from each donor to each recipient how many species were transferred. And you can see this really cool result that there's extraordinarily diverse outcomes from this. For instance, Taconic, terrible donor. Here we see that it transplanted 0, 2, 0, or 1 species to all these other mouse microbiomes. Jackson and Charles River were pretty decent, but this Envigo microbiome was extraordinarily good at transferring species to other microbiomes, transferring 33 to Taconic, 26 to Jax, and 23 to Charles River. Also interestingly, as a recipient, Envigo was very resilient to colonization by other microbiomes, except for this one species from Taconic that was able to colonize. And this was really cool because it told us that we had found a super colonizer microbiome, and we could start to unravel what makes a super colonizer a super colonizer. One moment, please. So one of our hypotheses was that these super colonizers were simply able to outcompete the recipient microbiomes through a faster metabolism, which we assessed by testing their ability to utilize different sources of carbohydrates. And we found that super colonizers from Envigo weren't better at metabolizing nutrients, but they could metabolize a broader source of nutrients. Here we see the Envigo in light blue could grow in all the possible sources of carbohydrates, including cellobiose and inulin, which the Jax microbes in orange could not grow on. And then very interestingly, after FMT, these communities in dark blue could now utilize inulin and cellobiose, telling us that these transplanting microbes are able to use these diverse resources. And this got me thinking that microbes might be super colonizers because they are capable of utilizing diverse nutrients, which led me to the key idea that colonization requires accessing nutrient niches within the host. And this has brought me to the conceptual framework I now use to think about microbiomes. So microbial ecosystems are like a puzzle, where each space in the puzzle represents a niche or ecological role within a community, and each piece represents a microbial species. To transplant microbes into these communities, we just need to get them to fit into one of these niches. So one way of doing this is by competing for a spot in the community. For instance, if you introduce a microbe that is more efficient at utilizing inulin, you can outcompete a native microbe and take its spot in the ecosystem. However, the microbes already present in the gut have an established foothold already and are generally extremely well adapted to their environments. After all, they've been with you your whole life. Therefore, generally competing for niches is not an effective route for colonization. A much more straightforward path is to simply access a niche that is not already in use. If no microbe in the ecosystem is able to utilize arabinogalactin, then it's very easy for an incoming species to fill that spot in the microbiome uncontested. Sorry. So my future research plan will leverage this conceptual framework to learn how to control the composition of microbiomes using high throughput approaches to engineer microbial communities in the gut. And as a systems biologist turned microbial ecologist, I have a unique interdisciplinary toolkit that will allow me to study these ecosystems at large scales that hasn't previously been done. In my graduate work, I implemented design, build, test, and learn pipelines using high throughput DNA synthesis and multiplex screens to dissect complex systems in a variety of organisms. Now, in my postdoctoral work, I've learned how to use cutting edge technologies to study how microbial composition changes over time and how the spatial organization of the gut microbiome changes, both across the length of the organ, as well as how these species are changing at micrometer scales. And my future research program will marry these two areas to create a new approach to studying microbiomes. In my first direction, I will explore using this principle of niche complementarity to facilitate FMT therapies. So in my postdoctoral work, we found that genetically identical mice from different vendors had vastly different microbiomes and niche profiles. So we will use a high throughput culturomics approach to profile the nutrient niches that each microbiome is capable of accessing, and then see whether we can use these niche profiles to facilitate robust colonization. Ultimately, we'll leverage metabolomics and metagenomics to build models that predict niche complementarity and use this to inform donor-recipient pairing strategies for FMT therapies. Our second direction will seek to develop next generation probiotics, where we will use culture independent and high throughput design, build, test pipelines to dissect the genetics underlying niche accession. So from this super colonizer microbiome, I've already identified tens of thousands of genes, which I call niche factors, that are putatively involved in metabolizing nutrients associated with colonization and found across diverse species. I will synthesize these using multiplex gene synthesis technology called DropSynth, and then engineer these niche factors into probiotic strains, such that I create a library of thousands to tens of thousands of engineered strains where each of them has a niche factor introduced to it. We will then administer this library to mice in multiplex all at once or in subsets and study how these different variants perform in the gut, both how long are they able to survive, are they able to find niches, and where and who do they associate with. And ultimately, this will allow us to comprehensively characterize the effects of these niche factors on colonization and provide us with genetic strategies to engineer probiotics with very defined functions. Altogether, this research program will improve our ability to manipulate the gut microbiome and develop bacterial therapeutics that can treat gut dysbiosis, thereby empowering us to enjoy even the most questionable food truck without fear. So thank you for your time, and thank you to all in the ISF community for making this such a wonderful conference, especially my mentors, Gladys and Jonathan, who helped me put my presentations together. Happy to take all your questions now. Thank you.
Thank you, Dr. Arteco, for your presentation. Really wonderful to learn more about your research. While we wait for questions from our audience, one question I had was how though the microbiome, as you shared, is very adaptive. How quick is the immune system to, and the gut, for example, to respond to adaptation of the microbiome?
That's a fantastic question. Yeah, so there's lots of context where the immune system will have to adapt to the microbiome. FMT is certainly for one. This is like introducing healthy bacteria, where typically it seems that with oftentimes when healthy bacteria are introduced, the immune system doesn't need to go through this learning process or anything. However, when pathogenic bacteria are introduced is a different story. So if you travel and you eat the wrong food or you drink the wrong water, you can introduce microbes that can aggravate the epithelial line, and then I understand that triggers the immune system and this learning process. But it's a bit out of my wheelhouse as far as the details of that. I'd love to collaborate with immunologists to know that role better.
Thank you for sharing. A question from our audience being, what is your plan for understanding the engineered factors efficacy?
Yeah, that's a great idea. The plan to understand the engineered niche factors efficacy in vivo, I will add. So the concept is that we can take these strains, give them a new gene, and then administer them to mice, and then see how this variant plus the new gene compares to the wild type probiotic strain. And because we can track these actually pretty precisely with next-gen sequencing technologies, we can see how the performance of these engineered strains compares to wild type, both as far as how long it survives in the gut. We can sequence different areas of the gut to see where they are colonizing. And then lastly, we can do these like high-profile spatial metagenomic sequencing technologies to see who they are associating with. So the objective will be to measure or take all these parameters and see how that compares to just the wild type strains.
Fascinating. We look forward to seeing your future research on the topic. And another question being, given the difference you saw in the microbiome of mice from different vendors, is this something that should be considered when thinking about selecting using mice for different disease models, microbiome being essentially involved in many different diseases?
Yeah, no, absolutely. Because, the role of the gut microbiome in all these diseases, I think is very unclear. But like people see mixed results, like, oh, the microbiome affected this disease. Other times it didn't. And I have to think about how much of that is due to just picking the wrong mice, ordering from the different vendors between institutions. So I think that is absolutely something that will affect a lot of these studies, but I don't think we know at this point necessarily what is the right microbiome to study these other diseases. I would say there are mice microbiomes that can better represent the Western gut. So maybe that would be a way to think about it. If you want a Bacteroides-enriched microbiome that represents the Westernized gut, then our results would say you should order from, I think that was Jackson has the most Bacteroides. But yeah, thinking about what population you want to best study and then what microbiome best represents that in mice might be the strategy to go around that.
Thank you for your response. On that note, what about the health benefits of the microbiome in general, but also in perhaps different disease conditions?
Sorry, could you repeat that?
Yeah, so also what about the health benefits of change of microbiome in general or in different medical conditions?
Yeah. Again, this is another thing that they see that it can play a role. Like certainly in the case of obesity with mice, there have been conflicting reports of whether or not transplanting microbiomes can be used to treat obesity. But given the link between the microbiome and diet, I think it has just the crucial role in anything related to cardiovascular disease or any metabolic disorders. The gut-brain axis and how it relates to neurological disorders is still a work in progress, but might also be important as we get more results.
Absolutely. One more. We have time for one more question. Our question being, what about the translation of your work for human use? Is that an area of interest for you?
I think that's wonderful. Studying the microbiome in the context of humans instead of mice. Obviously, that's the end goal. I think there's a lot of translational applications for this, especially studying how we can use colonization to stop pathogenics bacteria from entering us, or learning how the infant gut develops. I think these are very interesting areas. Harder to study, of course, because you don't have these very controlled models. But I think ultimately all of the insights we get in these very controlled settings, we're going to have to see if they can be replicated in humans as well. But very excited to do that in future research.
Thank you, Dr. Ortega, for your time and for sharing.
Thank you.
Thank you again, Dr. Ortecho. Really appreciated that talk. Okay, so next we'll have Dr. Joyana Gamble George. She is a postdoctoral fellow in neuroscience at New York University. And the title of her talk will be focused on multifaceted correlations of social connections. Dr. Gamble, I think you're muted.
So I think you can hear me now. So my talk is going to specifically look at HIV risk behaviors. And in terms of social connections, I'm going to focus on social capital. So for most of my research I'm currently doing is a little bit different from what I was trained to do. So I was trained as a cellular molecular neuroscientist. So when I was getting my PhD in neuroscience at Vanderbilt University, a lot of the scientific questions I was answering was very different. So as a pre-doc, I was interested in understanding more about mental health concerns in people. So we were trying to understand, well, how can we change a neurotransmitter system known as the endocannabinoid system and see if that is a potential way to come up with targets to help reduce anxiety and depression in people. But I wanted to switch gears a little bit after I left, finished my PhD program and went on to my postdoc work, first at University of Florida, because I found out during a lot of the research I was doing is that people that do suffer from mental health problems, they also suffer from substance use disorder. So an individual that might have increased anxiety, they might actually misuse substances as a form of coping. So as a postdoc, I wanted to understand more about drug-seeking behavior and then the behaviors that are associated with it that put an individual at risk for HIV. And then I wanted to get more into really understanding the social aspect of those at-risk health behaviors. Because a lot of the research I did as a pre-doc and during my first postdoc was in preclinical animal studies. So at NYU, I'm currently doing a lot of human studies and trying to understand those social and behavioral aspects of HIV risk behaviors and drug-seeking behaviors. So one question that I'm answering or asking is, does social capital play a role in HIV risk in people? So going on to the first set of studies that I did when I was a postdoc at UF, the reason why the research was so important was that there are a lot of people that are injecting drugs where they're sharing needles and then they're becoming infected with HIV. And then the interesting thing is that most of these cases are found in northeastern parts of the United States. one example state being New York. So in the lab of Dr. Habiba Kashbui at UF McKnight Brain Institute, we were really trying to understand what are those cellular molecular mechanisms or pathways that play a role in drug-seeking behavior, specifically in the context of people living with HIV. And we had this overall hypothesis is that when an individual share a needle where they're misusing drugs, and let's say they share it with somebody who's HIV positive, but they don't know, they become infected with HIV, we believe that the HIV infects the cell type known as microglia in the brain, and these lead to all these downstream signaling mechanisms that overall cause this hypodopamine state. So that means there's less dopamine in the brain or these neurons, as well as other cell types. And we think that what's going on is that there's a particular protein, the HIV-1 TAP protein, that is being upregulated and is entering into the microglia, and the microglia is releasing all these different factors like cytokines, reactive oxygen species that are playing a huge role in this hypodopamine state. And we think that because of this state, that is the reason why sometimes when people relapse and they go back to misusing substances, they're trying to somehow try to still seek that reward that they're getting from misusing those substances. But overall, we wanted to understand these pathways and mechanisms so we can come up with maybe a potential strategy for preventing this or treating this from happening. So there are a lot of brain regions involved in drug-seeking behavior, but I'm only going to focus on the midbrain and two regions within the midbrain, the ventral tegmental area and the substantial pars compacta. And that's because in that area, there are dopamine neurons, and dopamine neurons have been implicated in drug-seeking behavior and reward and motivation. So this is just a diagram of the animal model that we use for our studies when trying to understand drug-seeking behavior in the context of HIV infection. So this is a transgenic mouse model. And the really cool thing about this mouse model is that if you inject them with the antibiotic doxycycline, the mouse will actually express HIV-1 TAG in neurons and other cell types in the brain. And there's some other interesting features from an anatomical perspective as well as behavioral that you can find also in humans. So you will definitely find activation of microglia. If you don't know what microglia are, they're cells within your brain that play a role in removing a lot of the cellular debris in your brain. Also, you'll have structural changes in the brain. One example being reduced gray matter density. Then you'll find a lot of similarities in terms of behavior between this animal model and people, and that you'll find an increase in drug reward, even when a mouse is re-exposed to the drug. Then also you'll find increased anxiety-like behavior, and this animal model also have reduced social interaction. So one of the first questions we wanted to ask was what's going on in terms of the different cell types within the brain? How are they interacting or communicating with each other? So on the right-hand side, you'll see some images. These are with a confocal microscope, and in the green are the dopamine neurons, and in the red are the microglia. And what we're doing are measuring the interaction between the processes from the microglia with the dopamine neurons. And of course, we're doing this in the two brain regions that I talked about that are in the midbrain. I mentioned earlier, the substantial nigra pars compacta and the ventral tegmental area. And then on the left-hand side is a histogram, where what we found is that specifically in the substantial nigra pars compacta, you don't see as much communication between or interaction between the dopamine neurons and the microglia. So then our next question is, okay, we don't see a lot of communication between these two different cell types. So what is going on when you're activating the HIV-1 TAG in this animal model with those dopamine neurons? So on the right-hand side, you see again, some confocal images. And we're just showing the images of the dopamine neurons as they express tyrosine hydroxylase. These are in two different brain regions, the substantial nigra pars compacta and the VTA. And what we found, as you can see on the histograms to the left, is that HIV-1-TAT, when it's expressed in this animal model, we don't see as many dopamine neurons currently, like in the substantial negative pars compacta, as opposed to VTA. So something is going on at the protein level that we're seeing possibly cell death of these neurons. So then, since we do know that we're seeing changes at the protein level, our next question was, what's happening at the transcriptional level? What's happening with the mRNA expression? So we repeated our experiments again, but this time we're doing PCR experiment, where now we're trying to see how does HIV-1-TAT affects the tyrosine hydroxylase mRNA in the cells. And we didn't find any changes. So we believe that, yes, the cells are not communicating as much with each other. Maybe the microglia is releasing these reactive oxygen species such that it's leading to cell death in the neurons. But the changes apparently are occurring at the protein level, not the transcriptional level. So then our next question was, well, we know we're seeing all these molecular changes, but what's really going on with this cell in terms of this functionality? So then we did a lot of some electrophysiology experiments where we wanted to see the impact of HIV-1-TAT expression on the fire activity of these midbrain dopaminergic neurons. And this is with patch clamp electrophysiology. And what we found is that HIV-1-TAT, when it's expressed in the dopamine neurons, it does lead to a reduction in firing activity. So now we know that there's less communication between microglia and dopamine neurons. Changes are occurring that might be leading to cell death in the neurons at the protein level, because we're not seeing as much expression of tyrosine hydroxylase, and it's also causing a reduced firing activity in these same cells. So then after those experiments, when I left UF and I went to NYU, I really wanted to do more in terms of trying to understand what are the social factors as to why people are engaging in drug-seeking behavior that put them at risk for HIV. So at NYU, I collaborated with a social epidemiology lab at Yale School of Public Health to really try to understand the social aspects of HIV risk behaviors in people. So social capital, which I'm gonna get into, if you don't know what it is, it really refers to this umbrella term where it looks at interactions between people from a lot of different dimensions. So you might be looking at people's networks, the resources that they get out of these relationships, the value. It could be information, it could be finances, other forms of physical capital. It could be even social support. Then it gets into matters of people's perceptions of the relationships in terms of how much you trust someone. How much trust do you see within your community? Are your neighbors helping one another? Do you trust your neighbors? And also, are your neighbors participating in a lot of civic groups? So the reason why I focus on this research project is that there's not a lot of research known in terms of how does social capital affect people's decision to misuse substances or even engage in HIV risk behaviors. But for this talk, I'm just going to focus on the HIV risk behavior since we're limited on time. So we did a secondary data analysis where people that were living in the southeastern part of Pennsylvania, they were given this health survey, just asking them a lot of different questions about their health behaviors. One of them being HIV testing. They also asked them about their neighbors. How did they trust their neighbors? Did they interact with them? Did they feel like they helped one another out? So it got into a lot of different dimensions of social capital. And what we found after we did experiment correlation and a multivariable regression analysis is that the people who took the survey, the ones that didn't engage as much into HIV testing behaviors, actually had greater trust in neighbors. And we also found that this was very specific toward people that self-identify as Black or African American. So we assumed that from this result is that maybe those individuals, when they're maybe sharing needles with someone that is HIV positive, they become HIV positive, but they just don't know, maybe they trust that individual so much that they don't think they need to get tested for HIV. So that's why they don't go for HIV. Then you also, I think another assumption we have made about this or suggestion is that it could be based off of individuals' values and social norms within that community where they live next to their neighbors. And then the other interesting result is that we found something total opposite in individuals that identify as white, is that they engage in more HIV testing behaviors when they have a sense of belonging or connection with people in their neighborhood or community, or they feel that their neighbors actually are working together to improve their neighborhood. And this could be because of the social norms and values within that neighborhood. If they're so connected, working together, improve their neighborhood, maybe they have this sense of urgency or need to actually get tested to help to promote better health for people, including themselves. So you're probably saying, where am I going with all this? I did preclinical animal studies and now I'm doing human studies. So I did a lot of animal studies in the past that focus on mental health because I was really trying to understand the brain mechanisms that modulate these type of, mental health conditions, substance use, as well as associated HIV risk behaviors. And now I'm transitioning to human studies, where I'm getting into what are the factors that play a role as well in these behaviors. But ultimately, what I want to do is bring all of this knowledge, all of these experiments and these research projects together to have a lab that does both, where I can do the preclinical animal studies and the human studies and really understand the interactions between, brain mechanisms, genes, and social environment. And how does that impact the brain in the context of mental health and substance use and HIV risk behavior? So I'm hoping that within 5 to 10 years, I can answer these questions and ultimately take all of that information and come up with potential interventions that I can put out into the community that really help to address how do these, biological and social environment interactions really affect the brain? And how can it help to prevent or even treat people that suffer from mental illness, substance use disorders, or even behaviors that put them at risk for getting HIV. So I would just like to thank all my mentors and collaborators at the University of Florida, as well as at New York University and Yale School of Public Health. Do you have any questions?
Dr. Gamble, thank you so much for your fantastic presentation. Very much appreciate your interdisciplinary and different approaches for answering your research questions. While we wait for questions from our audience, one question I had is if there are potential differences when we consider aging as a factor in terms of the younger versus older populations with the research areas that you're investigating.
Yeah, so most of the research I've done so far is in older populations. So I think it'll be a little bit different with younger populations because one thing, the brain is still developing. It doesn't come fully developed until the age of 25. And then the other thing you have to think about all the influences, in terms of kids, adolescents, they're dealing with a lot of pressures within the home, then also in the environments where they're mostly at, like at school. So think about peer influences. So I think it will be a little bit different.
Absolutely. And on that note, you highlighted intervention and treatment strategies. Would you be exploring first through your animal models, exploring specific biochemical pathways in the context of potential intervention?
Yeah, so I want to first start with the animal models because it's just much easier trying to understand the cell molecular mechanisms. I think eventually I'm still going to do that in humans, but maybe do it with human-induced pluripotent stem cells that have been differentiated into neurons and then create like mini brains. But still, I think as far as, really trying to dissect things apart, animal models are much better. And then you can move and try to translate that information to human studies.
Thank you for your response. One question from our audience being, yesterday we heard a talk on the link between specific ethnic groups and cardiovascular disease. Based on your data set, do you have a way to suggest any social connections, ethnic groups, education, or accessibility to healthcare related to your research questions?
In terms of with a specific focus on CVD? So, okay.
That was the example given, so yes.
Okay. So I think because mine is getting into the social environment, so it does has a factor of social stress. And I know that stress in general, even social stress in terms of our interactions, depending on how we perceive those interactions, if they're very traumatic and we perceive them as negative, they can cause a lot of, I would say, symptoms that are on physical nature that can impact our health outcomes. So I think one of those being like CVD, or you're having hypertension, high blood pressure, maybe it might put you at risk for stroke per se. But I do think that there are a lot of correlations between the social environment, specifically stress, CVD, as well as some of the things that I'm looking at. And then also getting into the aspect of looking at this in terms of health disparities and underrepresented groups, where we know that those groups sometimes actually experience CVD at a much prevalent rate than other ethnic and racial groups. So I think those are very interesting correlations, and I know there's a lot to be done in that area.
Absolutely. Thank you so much, Dr. Gamble. We look forward to seeing your more findings from your fasting research in the future. Thank you for your time and for sharing.
Thank you again, Dr. Gamble. So this brings us to the close of our presentation session. It has been our pleasure to moderate today's session, and we want to give a big thank you to our fellows and the attendees for participating. We will now take a short 15-minute break, and the next session will start at 3:30 p.m. Eastern Standard Time for panel 4, which is Maximizing Community with Previous ISFS Fellows. If you have a moment, please take the time to visit the exhibitors page for our partnering institutions. Thank you.